Okay, so now look at this gene transfer. Okay, we start with gene transfer. So the idea of gene transfer, basically we take one gene. Okay, we take a gene. For example, in our study here, we use the insulin genes. What we're going to do here, we get the insulin gene from human. What we're going to do here is we transfer into another organism, the host cell here called E. coli. So the E. coli can produce the products of the gene, which is the insulin, and the entire process, we term it as gene transfer. Okay, we term it as gene transfer. Okay, so what we're going to do, okay, there are 5 basic steps in the gene transfer. First thing, we need to identify the gene of interest. So either we cleave it from chromosome, or we can synthesize it from mRNA. So this is for insulin, we use this method for insulin. Okay, so from mRNA, insulin mRNA, we're going to synthesize the insulin cDNA. Okay, cDNA. So once we get all these gene of interest already, we're going to insert it into a vector. So the vector here we're going to use here is plasmid. And then once we insert it, we need to ligate it, right? We need to ligate it. So again, we need to use the enzyme ligase. Okay. And then what we're going to do, because this plasmid, once they receive the insulin gene, they can't do anything. Okay, they can't do anything. They won't replicate. They won't be transcribed and translated. Therefore, no use. So what we're going to do, we need to put it into the host cells. So the process is called transformation. We transform into the host cells, but not all. The success rate is very, very low. Only about 1% of the host cell can take up the only 1%, okay, of the host cell can take up the plasmid. So we need to do the screening to make sure that the bacterial E. coli have successfully transformed. If they are not transformed, we don't want to culture them. We don't want, okay, to waste our money, waste our resources on the bacteria that cannot produce insulin for us. Once we successfully identify the the transformed, okay, bacterial, what we're going to do here, we can do the cloning. Okay, cloning here basically means that we clone the host organism. We give them nutrients, we give them a suitable environment, optimum growing conditions for them to grow, and then they multiply. So when they multiply, we have more. Okay, we do have more bacteria to produce insulin for us. Okay, so very quick ones. First, we need to isolate the gene of interest. Okay, so we need to prepare 2 molecules, 2 kinds of DNA molecule. Who are them? Gene of interest, in this case, insulin genes, and bacterial plasmids. And this bacterial plasmid is purposely, we purposely synthesize it. So this plasmid called PBr322. So what is so specific about PBr322 that we need to know? PBr322 actually contain origins of replication, contains this what we call the cloning site. Okay, this cloning site actually will be recognized later by the reaction enzyme called BAMH1. Okay, BAMH1. Okay, and then they also have this 2 genetic marker that allow us to screen. Okay, to do the screening later. Okay, so instead of cleaving out the insulin gene from the DNA in the relevant chromosome, we use mRNA for the insulins. So what? So this is very important question, highlight question always asked, why mRNA but not the DNA? Why we don't straight away use it? Why we need to isolate, okay, this mRNA coding for the insulins in the beta cells rather than the DNA in the beta cells? So the idea here is because insulin mRNA found in beta cells only code for the insulin, okay, while DNA has many genes that code for their respective protein, so more difficult to isolate. If you isolate this insulin gene, easier. Are you clear? Rather than the DNA. So for example, if we target the insulin mRNA, we can actually fish it out. We use the word fish it out or isolate out. Okay, we fish, something like fishing. Okay, we have the method to do this. But DNA, we have to search where. Only we can cleave it out. So more difficult. So second, there are large number of insulin mRNA molecule compared to the DNA, only 2 insulin gene on DNA molecule. One we get it from father, another one we get it from mother. Okay, and third one, very, very important. If you look at the insulin mRNA, they only contain coding regions, exon. But DNA contain both exons and intron, which if you incorporate it later into the bacterial E. coli, what will happen here is bacterial E. coli won't know. Are you clear? It's the language that's not understand by the E. coli. So E. coli do nothing. They don't care. They just translate everything. Then the amino acid sequence will be the wrong one. So we won't get the insulin at the end. Okay. So look at this. So how are we going to do this? We have to use the reverse transcriptase. Use the mRNA as a template. Use mRNA as a template so that, okay, the molecule can be converted to a double-stranded DNA molecule. We call it insulin cDNA. And here I want to introduce also, okay, the insulin cDNA can be synthesized by polymerase chain reactions, or just now I said PCR. So this part, no worry yet. This part we're going to learn it in PCR tomorrow. Okay, the next lessons. Okay, the next lessons we're going to talk about this PCR. At this moment, no need to worry yet. Okay, so what is the function of PCR? Understand in this case, this part, we want to make multiple copies. We don't want to have a small number. We want to have increase the number. So how to increase the number? The PCR techniques can help us to increase the number of copy. So PCR basically is a DNA replication. PCR actually similar to DNA replication. So our DNA replication in our body is, again, is inside the body, but this one we conduct it outside of our body. We conduct it outside of our body, okay, to make millions of copy. Okay, so once we are done already to get the molecule ready, now we're going to incorporate or insert the gene of interest into our vector. So what is our vector? We use plasmid PBR322. What we're going to do, we cut it with the registration enzyme. So highlight the registration enzyme that we need to know here, BAMH1. Are you clear? BAMH1. And we do know that yesterday we learned it. BAMH1 actually create the sticky end. Can you see that? It creates a sticky end. And if you look at this plasmid, PBR322, plasmid, PBR322 has this 2 antibiotic resistant gene. Okay, this is the first one. So the second one, antibiotic resistant gene. So these 2 antibiotic resistant gene act as a genetic marker. So we have the tetracycline resistant gene and also ampicillin resistant gene. Are you clear? So these 2 red color act as a genetic marker. Okay, they act as genetic marker. So no doubt, they also have the origins of replications, ORIs. Okay, origins of replication. So where is the cleaving site or registration site? Registration site actually located at this tetracycline resistant gene. So means that BAMH1 we're going to cleave. Are you clear? BAMH1 going to cleave here. So what happened if you cleave? So means that this plasmid, the tetracycline resistant gene no longer function. You try to imagine you break in between. Oh, tetracycline resistant gene no longer function, but ampicillin resistant gene still function. Are you clear? So BAMH1 going to cleave here. Later, we're going to insert our gene of interest into this tetracycline resistant gene. Okay, so if we use BAMH1 to cleave at the plasmid, then come back to our DNA molecule, our cDNA. Okay, insulin cDNA. So in order to create the same sticky end, right? Are you clear? I need the complementary sticky end, sorry, the complementary sticky end. We also use the BAM H1 to cleave it before and after the gene. So create the sticky end. Can you see that? Create the sticky end, create the sticky end. And these two sticky end actually complementary to the sticky end of the plasmid. Can you see that? So therefore, later, the sticky end, when they come to join together, then they can form the hydrogen bonds. Okay, they can form the hydrogen bonds, and then ligase will be able to seal the sugar phosphate backbones by forming the this phosphodiester bonds. So what we're going to do, we're going to mix this, the cleaved plasmid PBr322 and insulin cDNA are mixed together. And we do know that sticky end created by this BAM H1 will be complementary. So it can start to form this hydrogen bond between the complementary base pair. And then the insulin gene can be inserted into the tetracycline resistant gene. So understand the concept here. If I have the gene original, this is the part that students always confuse. If I have a tetracycline resistant gene, so you're going to give us the tetracycline resistant protein. So making the bacterial now resistant to tetracycline. But because we use the BAM H1 to cut this chart, so means that now if you insert the insulin gene, what will happen here is, okay, forming sticky end, and then with our insulin genes forming sticky end, and then continue with the part of the tetracycline resistant gene. So it becomes something like this. Are you clear? So we have to use a ligase to seal the sugar phosphate backbone. Understand? We seal the sugar phosphate backbone. So nothing already, done already. But do you realize that now tetracycline resistant genes no longer can be expressed already? We break it already. But insulin gene, yes. Insulin, yes, can be expressed. But tetracycline resistant gene no longer expressed already. So it means that this bacteria will be killed by the tetracycline later. So something like screening, can you see that? If we don't incorporate it, so it means tetracycline. If let's say BAM H1 cut it already and then religate, still remember yesterday we talked about religation because of the complementary sticky end, they can religate. We cut it, then they join it back. So in this case, they won't take in insulin gene. So how we know? So based on this, then we can screen out. So bacteria that don't have the insulin genes will be killed. Okay, so won't have the insulin genes will survive in the tetracycline. The one with the insulin genes will be killed by tetracycline. So this is a part of what we call the screening process. Okay, so question always asks why we need to use a BAM H1 to cut it at the tetracycline gene, because the bacteria that successfully take in the insulin genes won't be able to survive. Are you clear? And I know that most of you will have a certain question, why you want to kill them? They successfully take in already, why we kill them? So later I will explain it in the screening process. Okay. So this DNA ligase, okay, is used, okay, highlights, we need a DNA ligase now to seal the deoxyribose, or in this case, we can also claim it as the sugar phosphate backbone by forming this phosphodiester bond. So once you have done this already, when you have the plasmid with the insulin genes, this molecule is called as recombinant DNA, or called rDNA, or we can also call it as a recombinant plasmid because it's a plasmid. Okay, We also can call it as a recombinant plasmid. Okay, so ideally, okay. So in reality, we will say that in reality, PBR322 is an isolated copy of insulin gene are cleaved with the same restriction enzyme. We have to use the same restriction enzyme. So means that it will have the random mixture. What will happen here is, you look at this diagram. Okay, the one that successfully incorporated in the correct direction is this. Can you see that? All plasmid close back up. Can you see that? It close it back. Or the gene actually in the backward directions, which I don't want. These two I don't want. Okay, I only want this. This one is a success one. These two are failed. At this moment, don't waste time to go and check whether which one is correct, which one is wrong. Are you clear? Because we won't be able to check it at this moment, and the process is very, very tedious. So what we're going to do here is we put it aside. No problem. So means that if, oh, sorry. So in the mixture beaker, okay, in this mixture, so we're going to have two possible outcomes. First possible outcome is a success one. So this success we call as a recombinant. Okay, it's called recombinant plasmid. So this recombinant plasmid must be incorrect orientation. Okay, number two is unsuccess. Okay, so unsuccess one in this case includes the blank plasmid. Means that the plasmid, or we call unaltered plasmid, means that this plasmid actually don't have the insulin gene, or they have the insulin gene, but in reverse. Okay, in reverse orientation. Again, we don't want this too. But at this moment, in this mixture, what we can do? We don't want to do anything yet. We don't want to do anything yet. Are you clear? Okay. But to overcome, yesterday we talked about this, to overcome the reverse ligations, okay, or reverse what we call this gene orientations, we always can, okay. What we're going to do, we use two, okay. So cutting a DNA with two restriction enzymes simplify the situation because each molecule has a sticky end with different base sequence. So cleaved plasmid cannot rejoin back, and also isolated gene will ligate in the correct orientation. This, again, I put it here, the symbol indicates that for your informations only. And yesterday, I have shown to you guys, okay, I've shown to you guys how this reverse ligations won't happen if you have two set of, okay, restriction enzymes, okay. So in this diagram, you can see that, okay, in this diagram, enlarge it. So if I use this, can you see that two restriction, two equal R1 and not 1, okay, equal R1 and not 1, you can see that now this insert means the one that I want to insert it. What we're going to do here is, do you realize that here, equal R1s, then they are complementary. Here, not one, they are complementary. So means that reverse orientations won't take place. Because you reverse it, it becomes blue and red and blue. It cannot, because they won't form this, okay? They won't form what we call the hydrogen bonds, okay? So we come to the last part here before we take a rest. So how we can make this recombinant plasmid into the bacteria? So first thing, bacteria that must have the ability to take in this recombinant plasmid. So this cells is called competent cells. So how we make it competent? Okay, how we make competent? So we have two ways to make it competent. First, we can use the calcium chloride solutions. We give the cold and then expose to the heat pulse. Now heat pulse is not like heat short it for very, very long. It's just a few seconds. We heat short it for 30 seconds in this case, allowing to polarize the membranes, allows this plasmid to enter into the cells. Can you see that? So first, we have chemically competent cells. What we're going to do here is we incubate the cell together with the plasmid. We heat short it for 30 seconds at 42 degrees Celsius, and then the plasmid enter. Okay, So this is what we call the chemical transformation. Or alternately, we can use electroporations. Okay, we use the electric field to make the heat shots, electric shot in this case, to make a pore at the cell's membrane. Are you clear? So we're going to create a pore at the cell's membranes. So again, you can see that with this electric shot, then we will be able to get the plasmid to enter into the host cells. So when the host cell take it up, okay, so if you have the gene of interest, so insulin gene plus plasmid, we're going to get recombinant plasmid. And host cells with recombinant plasmid, this one we term it as a transformed host cell. So be careful when you use the word. Okay, it's called transformed host cells. Okay, but here, only a very small portions, only very small proportions of E. coli, around 1% is transformed, which take up the plasmid contain the insulin gene. So it's very, very small percentage. Are you clear? So that's why we need to do the screening. So what are the possible outcomes? So possible outcome, our syllabus, we're going to talk about, highlight here. First, we're going to talk about this, no plasmid. So means that we heat shot it, 99% won't have plasmid. Are you clear? Or you use electroporations, 99. This one almost 99%. Okay, around 99% or 98%. Are you clear? Okay. Then you can see that, okay, this is the one we're going to focus. Second, vector with no insert. Okay, why no insert? Why no insert? Because the religation of plasmid. So it means that they take in the plasmid, but the plasmid no insulin gene. Are you clear? And the third possible condition is a vector with desired insert. So it means that the inserts with the correct one. So this one is the recombinant plasmid with insulin gene. So our focus in our next part of the topics, next part of the topics, we talk about screening. We focus in these three conditions. First, no plasmid. Second, vector with no insert. So basically means that it's an unaltered, or later I'm going to call it as an unaltered plasmid. Mean the plasmid without insulin genes. And the third conditions here, vector with desired insert. So these three conditions, now your bacterial, 99%, almost 99%. This one, these two, almost 1 to 2% only. Okay, these two, almost 1 to 2% only. So how are we going to segregate them? How are we going to identify them? We need to do a screening process. Okay, we need to do a screening process. Okay, so we can see that the rest of the competent cells do not take up plasmid, or they take out the plasmid that do not contain insulin genes, or they take out the plasmids with the insulin gene but in the opposite directions. So that's why we want to do a screening. I don't want to waste my resources onto the bacterial. Okay, onto the bacterial, the 99% of them can't give me the insulin productions later. So that's why we need to do a screening. Okay. So with this, I end for the first part of the lesson.